Hello, and thank you and welcome to Asterias. Let me first start as an introduction to say we are the result of putting all of the stem cell assets of the Regeneron Corporation into Asterias. The first thing that we did with those assets, which included 12 years of research, a couple of $100 million that were spent, and all of the clinical and preclinical assets, and 400 issued and pending patents. So the first thing that we did is a prioritization process. This prioritization process was based first on probability of success, second on size of market and unmet need, and it had to have a significant healthcare cost associated to that market, no available therapies or adequate available therapies in the segment. And based on that prioritization process, and I forgot to say that we would be leading in this segment, we identified two platforms, and I emphasize the word platform because I'm going to speak each one having a clinical program, but with follow-on indications and potential areas to expand. The first platform is a pluripotent cell-based therapy. It is focused on spinal cord. It is focused on the cervical complete segment. The second platform is a dendritic, allogeneic dendritic cell therapy, and it is focused on lung cancer. I also want to say that both of the programs have very strong non-dilutive capital available to them. In the case of the spinal cord injury, we have been successful in securing a grant from the California Institute of Regenerative Medicine. It is for $14.3 million. It represents half of the cost of our program. For every $2 that we get $1 back. The second program has been partnered with the Cancer Research U.K. charity. This is one of the largest charities in the world focused on cancer, and this institution has conducted over 150 trials in cancer. It will actually produce all of the clinical supplies. It will carry on with all of the cost of the trial, and then we have the right to repurchase the data at a pre-agreed modest milestones and royalties. Again, this is a capital saving for us of about $25 million. So let me talk a little bit about the first program. This is one of the therapies that has been most studied. The FDA required us to do 28 studies and go to 3,000 animals. This is because this was the first embryonic stem cell product in humans. And I'd like to ask just to show you the two main aspects that were shown in this trial. Of course, it was safety, and we also showed, and you just saw the control group. Now you're seeing the treated animal, and you can see that the treated animal has recovered full mobility on both legs, both feet, while in the previous one it did not have it, and it can lift the tail. So we have seen very clear mobility improvements. We have also conducted a phase 1 trial with this therapy. This phase 1 trial showed that, again, it was safe. It did show initial signs of effectiveness. We have followed the patients for 4 years, and we can see a filling of the cavity where the injury occurred. We did not see mobility improvements because the dose that was used was very low, and that was imposed on us by the FDA. We are now moving into a phase 1/2A trial, and this is the trial itself. I don't have time to really expand on it. It will measure efficacy in the dose range of 10 million and 20 million cells, and the key or most important message of this, it will include 40 patients, which gives a very robust reading to position this therapy for accelerated registration. This is an example of there is an institution called SCOPE that has published how you can target the primary endpoints in this particular spinal cord subpopulation, and how it correlates to improvements in movement and also to cost. What this does to us is it provides a very clear path to market. Let me now move to the second platform and trial. This, as I already mentioned, it's an allogeneic dendritic cell. It is based on two previous trials that we call VAC1. They were also the same dendritic cells, the same telomerase as an antigen that we now provide through a cell line to the patient to make it allogeneic and be able to escape all of the issues associated with autologous vaccines. Okay, this is the example of or showing the trial itself. We are transferring and expect to complete the transferring of the manufacturing by the end of the month of June. Immediately, Cancer Research will proceed to produce clinical supplies, to get a regulatory clearance, and to initiate the trial. We are a public company. We are now valued at about $250 million. And we have, at this stage, I don't have time to expand on it, adequate cash between what we have on our balance sheet and the cash on hand. So let me just close here, and then we can see where you would like to expand. And sorry if I really rushed, but this 5 minute.
No, it's perfect, Pedro. Please come join me, and there's a microphone right behind your chair there. So, as you mentioned, I think Asterias is probably best known for the spinal cord program at this point, which I do believe was the first embryonic stem cell therapy ever to enter the clinic. So there's really a lot of experience with these cells in man for an embryonic stem cell therapy. You mentioned the phase 1 data on the first 5 patients. Maybe you can just go into a little bit more detail on what that experience taught you. I know it was at a 2 million cell dose in the thoracic region. So maybe you can tell us why the FDA asked you to start there and sort of what your learnings were from that initial exposure in man.
Well, before I do that, I do want to say that one of the things that we have done is we have rehired the key scientists from Geron so that we would bring not only the assets, but the knowledge not only of what works, but also of what didn't work over those 12 years. So the answer of why the thoracic area has a longer area for the cells to travel, and it is in an area where the probability that the cells might escape into vital organs is low. So the FDA requires for all cell-based therapies to start in the thoracic area, prove safety, and then move on to the cervical area if that is the targeted subpopulation. So that is the primary reason.
Now, in the ongoing study now, and I'm sorry, in that phase 1 study, you were actually able to show, though, not only the safety of the cells, but also some morphology and some histopathology, well, not histopath, but some clinical benefit in terms of the cells really filling these gaps. So maybe you can tell us what you did see with respect to what the cells actually accomplished in those lesions.
Well, the first comment is in the 28 trials we did with the rats, we knew that the dose level that we were using in thoracic area was not sufficient to show mobility. The equivalent dose in the rat was 60,000 cells, and that did not show mobility, so it was not surprising to us that that did not happen. What we did see is, and we have now followed those patients for 4 years, is that there is a filling of the cavity at the site of the injury, and that that filling has remained through MRI. Obviously, we cannot do biopsies through the 4 years. So it's not signs of mobility, but it does show that the cells are there and the cavity is filled, which is not the case in historical controls.
And they're engrafting. Now you're conducting a larger phase 2 study, and you've moved up to the cervical region, cervical injury patients. Why are you required to start at that same 2 million cell dose? And tell us a little bit about how this adaptive trial design works with this phase 2 study. How does the different cohorts start?
Well, again, the FDA believes that when you move to the cervical area, you have a shorter area to travel for the cells, and that the cells, in this case, can escape to the brain or to other vital organs. So they require us to do the first 3 patients at the same low dose that we did in the thoracic area. We do not know exactly how the efficacy will perform because now we have a shorter area to travel, and we might be surprised, although we're not counting on that. Rapidly after the 3 patients, we will be moving to the first efficacious dose at 10 million cells, 5 patients. And as we observe the mobility improvements of that group, we will then, and this is where the adaptive design comes in, we will then decide whether that is most likely to be the targeted dose to go to market. And if it is, we have 27 more patients that we will add to that cohort, while in parallel we do the 20 million cell. Now, the objective of having so many patients at a phase 1/2A is to position the therapy with a robust reading of efficacy so that we can be a very good candidate for an accelerated regulatory approval and move directly to pivotal trial.
And what would be some of the primary efficacy endpoints that you'll assess in that study?
Again, one of the primary reasons why we chose cervical complete is because this external group that I mentioned has clearly published what is the spontaneous recovery of this group, and it's a very homogeneous subpopulation. So what we expect is to double the spontaneous recovery, which is in 20% of the patients, we see 2 levels of mobility in the cervical area, which might mean that you now control your bowel movements, etc. The table I showed actually shows that. We will be now trying to look for 40% of the patients showing that effect.
That's helpful. Since we have about 3 minutes left, I want to transition to your other area of research in terms of immuno-oncology. And VAC1 was the first product that you developed, and this was an autologous cancer vaccine. So firstly, maybe tell us why using telomerase, and what did you learn from your initial clinical experience with VAC1?
Well, first, the reason why we're using telomerase first is because Geron has owned telomerase for a long time. What Geron is targeting is very different from what we're trying to do. Geron is actually targeting telomerase itself. We're using telomerase to create an immunity response. But having said that, our scientists know telomerase up and down. And also there is a lot of literature that clearly shows that telomerase is key in cancer. It is present in about 90% of cancers, and there has been clear immune responses reported on it in several cancers, including our 2 studies. So our 2 studies were conducted in prostate cancer as well as in AML. Both showed, of course, safety, but both showed efficacy indicators. Geron stopped it at 1 year, so we were not able to follow at that time the patients until we would see an overall survival rate, which, by the way, we're now doing.
Great. And now, as you mentioned in your slides, you're actually developing an allogeneic version called VAC2 and have partnered with Cancer Research U.K. and should actually be starting that study, I think, late this year, early next year. So how do you anticipate kind of rolling out data from that study, and what is your retained interest with respect to the product that's being developed at Cancer Research U.K.?
Well, first, we have a series of intermediate readouts of both studies. In the case of the VAC2 program, our first will be the full transfer of the technology, then it will be regulatory approval, followed shortly by the initiation of the trial. Then, of course, in that trial, there is also a safety cohort. Then there is 2 cohorts in advanced disease, in lung cancer, and then also in resected disease.
Excellent. Great. Well, with that, we're just out of time. And I want to thank, firstly, Pedro for joining us for our presentation from Asterias. I also want to thank you all for joining us for the end of the day. It's been I hope that you found today to be informative and worthwhile. I know that I did. I want to again thank the organizers, the Alliance for Regenerative Medicine, as well as all of the sponsors, my bosses at Piper Jaffray, Edison Research, Cryoport, CTI, Lonza, Kawasaki, Neostem, PCT, Traxel, and PCG Advisory group. I'd also like to thank both Malcolm Brenner and Olivier Danos for speaking with me this morning at the opening panel. I'd like to thank my partner, Josh Schimmer, and also my good friend, Jason Cobert, all of our panelists for such interesting discussions throughout the day, as well as Jim, Tony, and Carl for their talk over lunch, as well as all of our company managements that presented throughout the day. I think you'll all agree that today was even better than last year. Like I said last year, and I'm going to echo it again this year, there really is a revolution that's taking place in the fields of cell, gene, and regenerative medicine. And I expect continued clinical data, product approvals and launches, as well as an increasing number of high-profile partnerships and acquisitions in the coming 12 months to continue to drive performance of the sector. I get asked all the time, are we in a biotech bubble? And while today's sell-off may be spooking some people, and I think that there are maybe some segments that are overheated or individual names that are overvalued, it's really the fundamental innovation that is coming out of this biotech sector, and I think led by these fields of cell and gene and regenerative therapy that are going to deliver continued growth over the next several years to come. So I want to thank you again for all joining us. It's going to be an exciting field to continue to watch, and I hope you'll be able to join us again for next year for the 4th annual ARM Regen Med Investor Day. Thank you very much.